Hello everyone. This is my first video on YouTube, and I'm currently a second year Mbbs student. So today I'm going to discuss about this topic of inflammation on pathology. And first I'll talk about acute inflammation, and I will cover this topic of acute inflammation in two videos. So this is the first video actually. So basically inflammation, as you can see, it's a non-specific response of our body. Okay, it's not a specific, it's a non-specific response of our body. It's basically a protective response by our body. So this inflammation can be of two types, this acute and chronic. So acute is, as the name suggests, it's short-lived and usually stays for few weeks or few days. And on the other hand, there is chronic inflammation. This chronic inflammation is long-lived and lives for months, years, or maybe decades. So what are the mediators of acute? Means the principal cells, it's the neutrophils are the principal cells of acute inflammation. And for as far as the chronic is concerned, the macrophages are the principal cells. And so neutrophils for acute inflammation and macrophages for chronic inflammation. And so the signs, what are the signs of acute inflammation? So there are four cardinal signs of acute inflammation. First is the rubor or the redness, the calor or increased temperature locally at the site of inflammation, and then comes the dolor, pain, and then lastly comes the tumor or swelling. So four cardinal signs basically: rubor, calor, dolor, and tumor. There is another one also. There is functionalisia, which is loss of function. So in case of for example, in case of acute viral hepatitis, there is loss of function of the hepatocytes, which causes jaundice. So these are the cardinal signs. So this is the fifth additional point. So functionalisia is also actually cardinal sign of acute inflammation. And for chronic inflammation, the chronic inflammation gradually heals by fibrosis. There is no regaining of function actually. In chronic inflammation, there is no regaining of function. So, and the organ will be shrunken. The organ which is affected will be definitely shrunken, and there will be induration seen. There is characteristic induration which will be seen in case of chronic inflammation. Induration is basically the hardening of the organ or tissue which is affected. And there will be function loss. So as in case of acute inflammation, there will be functional issues. Also there will be function loss in chronic inflammation, but in chronic inflammation the thing is it will be a permanent loss of function. In acute inflammation there is no permanent loss of function, but in chronic inflammation there will be permanent loss of function. So these are the basic features of acute and chronic inflammation. This is a basic overview which I have given you. And acute inflammation generally heals by regeneration, but this is not the case for chronic. For chronic there will be permanent loss of function, as I have said. So now I will come to the mechanism of acute inflammation. So the mechanism of acute inflammation is generally very important for your professional examinations, university examinations, and also for your NEET PG. Okay, so there will be two events, basically the cellular events and there will be the vascular events. So first the cellular events. Cellular events is basically a little bit complicated. Okay, it's a little bit complicated, but let's discuss this. So first we encounter an acute inflammation. Okay, then the neutrophils will be brought to the site of inflammation. Okay, so the neutrophils, they circulate in the blood vessels, so they have to come to the site of inflammation. So neutrophils, which are actually present in the center of blood vessels. Okay, the blood cells in the blood vessels move in a laminar fashion. Okay, so the neutrophils will be present in the center of the blood vessel. And there are basically two types of pools. First is the circulating pool, and another is marginating pool. The circulating pool basically keeps on circulating, and the marginating pool, which is attached to the endothelium. The marginating pool, as you can see here, it is attached to the endothelium. And the circulating pool, it keeps on circulating. So these neutrophils, 50% of these neutrophils are in circulating pool and the rest are in marginating pool. So 50% neutrophils in circulating pool, 50% in marginating pool. Okay, so neutrophils they have to move from the center of blood vessels to the periphery, because as soon as we encounter an acute inflammation, there will be loss of this laminar flow. As I have said that blood cells usually move in a laminar fashion. Okay, so neutrophils are basically present in the center. The neutrophils have to come from the center of the blood vessel to the periphery. So there will be loss of laminar flow, so the neutrophils will have to come to the periphery. So the first step which occurs is leukocyte margination. So first step in acute inflammation is margination. So leukocyte margination is basically what I am trying to say, movement of leukocytes from the center to the periphery. Why? Basically due to loss of this laminar blood flow. So loss of this laminar blood flow will lead to leukocyte margination. This is the first step. Okay, first step of this acute inflammation. And see the second now. So the margination happens, so neutrophils will have to start attaching to the endothelium. Okay, so neutrophils start attached to the endothelium gradually, but this attachment will be a weak attachment. Okay, this attachment is not so strong. The first initial attachment is weak attachment. And neutrophils basically they get attached, then they get detached, and again get attached, so on. So this is not a strong detachment, you can say. This is a weak attachment. So this will be the first step. Next comes the leukocyte rolling. So why we are calling it rolling? Basically as if, so imagine as if the neutrophils are rolling over the floor of the endothelium. Okay, so this is the endothelium. Basically this is the endothelium, and neutrophils are basically rolling over this endothelium. So the second step is called leukocyte rolling. Okay, so first is margination, second is leukocyte rolling. So this rolling is mediated by selectins. Okay, the selectins are present on the neutrophils, which does the job of binding to the endothelium. Okay, this rolling is mediated by selectins. Okay, so after rolling, as you can see that, as you can understand that this rolling is not a strong attachment. So it keeps detaching and it keeps moving forward, the neutrophil. So we have to arrest this flow of neutrophil, because neutrophils have to be recruited to the site of inflammation, right? They have to be brought to the site of inflammation. So we need a strong attachment. We can't have this weak attachment. It won't do our job. So for strong attachment to the endothelium, it is done by the help of integrins. Integrins are basically present on leukocytes. They bind to the ligands which are present on the endothelium. So this step is called leukocyte adhesion. So leukocyte adhesion is a strong attachment, not a weak attachment, unlike I told about margination and rolling. So this leukocyte adhesion is a strong attachment. So this leukocyte adhesion is done by integrins basically. So important ligands, I was talking about the ligands present on the endothelium. So important ligands are cellular adhesion molecules, or CAMs, which we say. So the cellular adhesion molecules are the important ligands which are present on the endothelium. So the integrins in the leukocytes will bind to the ligands which are present on the endothelium. Okay. So what are the important cellular adhesion molecules? It is very important, we need to know. So first is the ICAM, VCAM, and the PCAM, which is basically CD31. So ICAM, VCAM, and PCAM, these three are important. These we have to know basically. These are the important cellular adhesion molecules which are involved in this process basically. So this CD31, this PCAM which I am saying, CD31 will activate the neutrophils by binding to the PCAM. The neutrophils will release the granules. Important granules are released, which I am going to talk in a short while. And important cytokines are released as a result of two of them are IL-1 and TNF alpha. So okay, the IL-1 and TNF alpha, the important cytokines which are released. So these two will lead to endothelial injury. Okay, so these two will lead to endothelial injury. This IL-1 and TNF alpha is very important, which leads to endothelial injury. So why this endothelial injury is important? This is basically important for further events to go on. Okay, so this is basically leukocyte mediated endothelial injury, not only endothelial injury, leukocyte mediated endothelial injury. So basically what happens, this strong binding by the integrins, important cellular adhesion molecules are released basically, and the CD31 basically activates the neutrophils, granules are released by the neutrophils, and IL-1 TNF alpha leads to endothelial injury. Okay, so what are the 5 events? First is the margination, then rolling, then adhesion or integrin binding. Okay, then will be the last two processes will be the diapedesis and chemotaxis. Okay, diapedesis and chemotaxis I will come in a short while. And So first, what I have said that neutrophils release granules. Okay, so the release of granules, two of them I have already mentioned, IL-1 and TNF alpha. This IL-1 and TNF alpha will lead to endothelial injury. This endothelial injury will lead to increased vascular permeability. Okay, so increased vascular permeability is very important in this process. Okay, I will explain it in a short while why endothelial injury is important. And also some important enzymes are released. One of them is collagenase. Collagenase, to be more specific, it's type 4 collagenase. In basement membrane, the collagen which is present, it disrupts the collagen of the basement membrane. So basically leads to disruption of the basement membrane, and it leads to multiple perforations in the basement membrane. And these integrins, I was saying, integrins will bind to PCAM, which is CD31, which will lead to release of IL-1, TNF alpha and important granules, and leads to endothelial injury and increased membrane permeability. So this collagen leads to membrane disruption. Holes are created, several holes are created, so the neutrophil can come out of the perforated basement membrane. Okay, so they can basically squeeze out of it and come out of the vessel. So this is an important step. This is known as diapedesis. So for diapedesis to happen, we need increased vascular permeability and we need this perforation in the basement membrane. This job is done by the collagenase. So neutrophils release granules. These granules will cause perforations, and through these perforations, neutrophils can come out. So neutrophils can squeeze out of these blood vessels and this basement membrane basically, and they will lead to diapedesis. And as you can see, they generally squeeze out like this. The neutrophils can cross the basement membrane from inside to outside. So the movement is from inside to outside, not outside to inside. Okay, inside to outside of the blood vessel. So this is diapedesis or transmigration. This is basically very important. Okay, diapedesis process. This is an across movement. Okay, so diapedesis occurs only in the venules, but in exception is in lungs. In lungs it occurs in the capillaries. So diapedesis mainly occurs in venules, but in lungs it occurs in capillaries. Okay, this is an important point to note basically. So the movement of neutrophil from outside the blood vessel to the site of inflammation is the last step, which is called the chemotaxis. So after diapedesis, the blood vessel with the neutrophil which comes out of the blood vessel must be recruited to the site of inflammation, right? So this chemotaxis is basically tissue migration. Okay, so chemotaxis is the final process of acute inflammation. Okay, so see, this is chemotaxis. So chemo means chemical and taxis means movement. So movement because of chemicals. So chemotaxis is basically movement occurring because of chemicals, and this chemotaxis is mediated by chemotaxins. Basically, chemotaxins are chemicals which induces this process of chemotaxis. The examples are important: IL-1 and TNF alpha, which is produced by neutrophil and released in the blood vessel. These are important chemotaxins, by the way. The second is the complement C5a. It's also an important chemotaxis. We will get to know about it shortly. Then comes the leukotrienes. Among the leukotrienes, LTB4 is an important chemotaxin. And also chemokines. Chemokines are basically cell-specific chemotactic agents. So chemokines are also important chemotaxins actually. So this IL-8 is present on the neutrophils. This eotaxin is present on eosinophils, and these two are important chemokines actually, which help in chemotaxis. We will discuss these things shortly. And finally, there are some bacterial products. So all these things, this complement, leukotrienes, chemokines, bacterial products, and the above mentioned IL-1 and TNF alpha, these are important chemotaxins which help in chemotaxis. So all these are basically proteins secreted by many cells in the blood, right? So all these proteins will have to come out of the blood vessel for chemotaxis, right? So all these proteins we need to come out of the blood vessel. So blood vessel is basically, it's, as we all know, it is physiologically not permeable to proteins, right? Blood vessel is not physiologically permeable to protein, so we need to increase the vessel permeability, right? So vessel permeability should be increased before chemotaxis. You have to note this point. You have to note this point that before chemotaxis to happen, the vessel permeability must increase. So we need to increased vessel permeability for chemotaxis, or the final step of acute inflammation to happen, right? So this is the final step of the events of, I mean the cellular events. Next comes the vascular events. So see the vascular events. For the vascular events, chemotaxis, before chemotaxis, increased vascular permeability is very important. So the vascular permeability must increase before chemotaxis to happen, right? So the increased vascular permeability is a very important thing for chemotaxis to happen, and this is the most characteristic feature of acute inflammation. So I will cover it in a separate topic. Okay, so this is all for today. I have mentioned a lot of important things basically. So basically I have acute inflammation, I have talked about the cellular events. So cellular events I have completely covered. So these things are actually very complicated in Robbins. If you see, these topics are given in wide detail. I have tried to cut short and be precise, as much precise as I can, and I hope you guys enjoyed this video, and thanks for watching, and more videos will be coming and I will be working on it. So thanks for watching.